Platelets are one of the three main types of cells that we've talked about within blood. And I must clarify at this point that platelets are not true cells. They're enucleated and they're really just cell fragments. And red blood cells, erythrocytes, are not true cells because they are enucleated. They have no mitochondria. And as we talked about, they're just bags of hemoglobin. So typically, instead of referring to blood cells, which would include white blood cells, red blood cells, and platelets, we tend to call them formed elements because the latter two, red blood cells, otherwise known as erythrocytes, and platelets, are not true cells. So platelets are involved in hemostasis, that is the cessation of any sort of hemorrhage or bleed within the body. So let's take a look at this. In this picture, what we have is a blood vessel, and we've talked about blood vessels in previous videos, but I just want to outline some regions of the blood vessels. So right in here would be the lumen where blood is passing through. Lining the lumen are simple squamous cells known as endothelium. That is the lining of the blood vessel. Immediately outside or above in this picture in blue, and below the endothelium here in blue, is a collagenous layer. It's a tissue layer predominantly formed by collagen. And then let's suggest the outermost layer, the red, is smooth muscle. There'd be another layer of connective tissue on the outside of that, but we're not concerned about that at this point. So during a hemorrhage, what happens is we get some sort of laceration in the blood vessel. Keep in mind, this could be within an intact body, say a blood vessel that ruptures inside your body, or it could be on the outer surface of your body where you're losing blood to the environment. But in this case, what has happened, if we look right here, there's been a disruption in the tissues and cells in this specific region, specifically in the endothelial cells and in the collagen. What happens in a hemorrhage, it exposes this collagenous layer to all of this blood right here coming through. Normally, the only layer that's exposed to blood is the endothelium in green here. But now the blue layer is exposed to the blood. So the problem is we have a high pressure system initiated at the left ventricle that's pushing blood throughout the body to the periphery, to the vital organs. And that is still happening until something is done. So blood is just going to keep oozing out of this laceration until something is done. So the first thing that needs to be done is vasoconstriction. Vasoconstriction just upstream of the laceration. And let's just keep in mind that we're moving from left to right. That is to say, the left ventricle would be upstream this direction, and the tissues that this blood was originally trying to access would be down here on the right. So as blood is moving from left to right, vasoconstriction is going to limit the passage of blood through this area. If the hemorrhage is significant enough, theoretically, it could cut off the entire blood supply downstream of this area. If nothing else, the goal is to minimize blood flow to this area, not totally cut it off, and I'll highlight why that is shortly. But by vasoconstricting this area, we have decreased the volume of this blood vessel, and by decreasing the volume, we've increased the pressure, which decreases blood flow through this area. Now, certainly, we still want blood flow through this area because there are constituents, components of the blood that are going to help us clot this blood vessel. And the first one of those are going to be platelets. Platelets are going to arrive to the area and immediately stick to the collagenous layer. This platelet plug happens because these platelets are attracted to the collagen fibers, somewhat like a magnet. They just start sticking to the collagen and stick to each other to start forming the initial plug. In an intact blood vessel, say in this region or here or anywhere in the body, platelets are constantly flowing through the blood, but they don't stick to the innermost lining, the endothelium, because the endothelium secretes some signaling molecules that actually deter platelets. One of those is nitric oxide. But here, where the laceration has occurred, because there's no endothelial cells, there's no repulsion of these platelets. The platelets come in, start adhering to the collagen fibers, and initiate the platelet plug. During this time, the platelets are going to secrete platelet activating factor, which is going to attract more platelets to the area. When more platelets get to the area, they're going to secrete more platelet activating factor, which will attract more platelets to the area. This is a classic positive feedback loop. In addition to this, these platelets are going to secrete serotonin and thromboxane. Truthfully, it is thromboxane A2, but for our purposes, let's just say it's thromboxane. Both thromboxane and serotonin are going to help maintain and even increase the vasoconstriction that initially happened. Once that occurs, what we start getting is a full-on blockage of that laceration, and that is aided by fibrin. Fibrin is a protein that helps the platelets stick together and ensures that no blood loss is going to occur. So fibrin along with the platelets are necessary for a complete cessation of the bleed. Now, fibrin is an active protein. Its precursor is something we've already talked about, and that's fibrinogen. And we talked about it when we were talking about plasma proteins and colloid osmotic pressure. We don't always want fibrin flowing through the blood, because if it did, it would cause platelets to stick together, and we may get a thrombus or some sort of clot throughout our vascular system when we don't need it. So we do not always want fibrin active. Fibrinogen is an inactive protein, and it only gets converted to fibrin in the presence of the enzyme thrombin. So thrombin is the enzyme that converts fibrinogen into fibrin. When that happens, we get a repaired blood vessel. So there's a couple of things that are involved in repair the blood vessel. First and foremost, we need to get rid of that clot that stopped the cessation of blood, because what that clot did is it narrowed the lumen of the blood vessel. We want to get rid of the clot and return the lumen to its normal size so we can have our normal blood flow. And that's achieved by another protein that has to be activated, known as plasmin. Plasmin is a protein that breaks down that clot, dissolves the clot. But that comes from an inactive precursor, another plasma protein we talked about, plasminogen. Plasminogen is an inactive plasma protein, but in the presence of another enzyme, tissue platelet activator, it gets converted to plasmin to dissolve that clot. One thing I haven't discussed in this whole process is growth factors that are needed to repair the collagenous layer and the endothelial layer. But for now, that'll be the extent of hemostasis.